So it's my pleasure to introduce our final speaker of the session, Itzel Meneses from Universidad Nacional Autónoma de México. And she'll be talking to us today about the systematic modification of a FRET biosensor based on an intrinsically disordered protein for trafficking the effects of osmotic stress in Arabidopsis. Awesome. Thanks for joining us.
Thank you. Thank you very much, Margaret. So do you see my screen?
Yes, it looks great.
Thank you. Good morning, everyone. My name is Itzel Meneses. I am a master's student at National Autonomous University of Mexico in Cuevas de la Ciudad. Today, I'm not trying to convince you that plants are exposed to external environmental conditions. However, dramatic shifts in the external environment cause intracellular effects, change in the cell. One of the main barriers to better understanding the intracellular effects of osmotic stress is the lack of methods that dynamically monitor changes in the physicochemical properties of single cells, non-destructive manner, and in real time. Nowadays, we know about the genetically encoded fluorescent biosensors that are optimal tools that enable the dynamic visualization and quantification of biochemical effects. So, in order to understand this, our lab designed and developed a FRET biosensor based on an intrinsically disordered protein from Arabidopsis thaliana called late embryogenesis abundant proteins or LEA proteins, expressed under water deficit. Our biosensor named SED1 utilizes a LEA protein from group 4 as a sensory domain fused to a FRET pair, M-cerulean 3 as a donor, and citrine as an acceptor. LEA-4-5 exhibits a reversible disorder to folded transition in response to increased osmolarity and or molecular crowding, allowing approach between the fluorescent proteins, causing FRET phenomenon. SED1 track changes in osmolarity in a wide set of organisms, E. coli, Saccharomyces cerevisiae, human cells, and Nicotiana benzamiana. However, the major limitation that SED1 presents is the inability of reporting the osmolarity changes in Arabidopsis thaliana. In the next essay, I show you that when we express SED1 in nuclei of Arabidopsis thaliana before and after osmotic shocks, we did not observe FRET change. So SED1 is not functional in Arabidopsis thaliana. We think that since LEA-4-5 is an Arabidopsis thaliana protein, the lack of response could be its interactions with endogenous partners and or post-translational modifications. According to this, previous research has shown that LEA proteins at almost all serine, threonine, and tyrosines are hyperphosphorylated in Arabidopsis thaliana. So we might that if the introduction of negative charges throughout the protein could modulate the structural transition of the sensory domain of SED1. Another possibility of its lack of response is the donor fluorescent protein M-cerulean 3 that experience fast photobleaching in plant cells. So, in order to approach this hypothesis, our lab, we propose to systematically modify certain characteristics of SED1 to enhance versions that could be functional in Arabidopsis thaliana. Here I show you our new versions. A first version incapable of being phosphorylated SED1 phosphoryl. This version, all the serine, threonine, and tyrosines of LEA-4-5 were replaced for alanines. Then, when we express this version in these cells and then test the biosensor response, we observe that SED1 phosphoryl exhibits lower FRET radio change than SED1. Also, we notice that the G cells have a growth rate of has a growth rate of cells suggesting that Z1 phosphoryl is toxic for the yeast cells. In accordance with this, Z1 phosphoryl did not accumulate in nicotinic transient expression. On the other hand, we have the second version, a version with a different pair, Z1 M turquoise 2 M neon green. This version has a new Fred pair, M turquoise 2 M turquoise 2 as an donor and M neon green as an acceptor, keeping the allele for 5, the sensory domain of Z1. Then we test the biosensor response under hyperosmotic stress of Z1 M turquoise 2 M neon green between Z1, and we observe that Z1 M turquoise 2 M neon green exhibits a Fred radio chain comparable to Z1. Contrary to Z1 phosphoryl, Z1 M turquoise 2 M neon green accumulates in nicotina ventamiana transient expression. And to test the biosensor response in nicotina ventamiana leaves, we took a small disc of nicotina ventamiana leaves and placed onto a small disc, a plate with wells containing hyper and hypoosmotic solutions. As a result, we observe a typical Fred behavior in M turquoise 2 M neon green with an increase of the fluorescence intensity and with a decrease of the fluorescence intensity of the donor. According to Z1, that is, these results was previously published in the paper. Unfortunately, I don't have data from the biosensor getting Arabidopsis thaliana. However, we are currently getting homozygous Arabidopsis thaliana transgenic lines. I will be glad to share these new results in the next speak. In conclusion, to sum up, Z1 phosphoryl is toxic in this cell and it is not expressed in plant cells. Z1 M turquoise 2 M neon green shows a comparable Fred response to the original Z1. And we are currently generating Z1 M turquoise 2 M neon green Arabidopsis homozygous transgenics lines to test the biosensor response. Finally, I would like to mention a project carried out by a graduate student, Constanza Enriquez, about 200 biosensors based on IDPs of different organisms. She expressed this biosensor library in these cells under hyperosmotic shocks to test the Fred change. As a result, there are biosensors with high improved sensitivity. And hopefully with these new biosensors, the next step is to express this in Arabidopsis thaliana. Lastly, I want to thank al Departamento de Bioquímica de la Facultad de Química de la UNAM, the funding, the collaborators, and the lab, Cuevas Velázquez. I will be glad of answering any question. Thank you very much.
Really cool talk. Thank you so much. I was curious for your first version of the Arabidopsis SED1 biosensor test where you couldn't see a response. You were showing root cells. Do you think there's any like cell type specificity to seeing the response? Like all the previous assays were transient expression in leaves. Do you think that there's a difference in the response that you might see in root cell types versus leaves, or do you think the cells in terms of osmotic stress would behave really similarly?
I think it's going to be similarly in the expression of this biosensor. You referred to Z1 phosphoryl?
Well, yeah, I mean, I was just thinking about because you had images from your transformed Arabidopsis line, but like in any of these versions of the osmotic sensor, once you have one that is stable in Arabidopsis, hopefully this updated version that you're transforming, do you think that like the root versus leaf cells would respond equally in terms of giving you osmotic stress data, and like the sensor would work equally well in those different cell types?
Yeah, I think it would be, we have difference, but yeah, I would think that.
Cool. Yeah, it's a really neat tool that you're developing. Let's see. I don't see a question in the chat, so I'll ask one more question and then provide transition slides for our community discussion that's going to start at 8:40 Pacific time, 11:40 Eastern time. I thought the orthogonal biosensors across species was really, really cool. What was the one that was like super responsive to osmotic stress? There was like one line that like was way above the other response.
Sorry, in this one?
Yeah.
Yeah, this one is an IDP from tardigrade at Cache.
Awesome. Cool.
Yes.
Yeah, that'll be very cool. Thank you so much. Oh, there's one more
Thank
question
you.
in the chat.
Okay.
So this question is, do you think this biosensor is only specific for osmotic stress? So that's a good, yeah. So is said one only reporting on osmotic stress?
The finally of this biosensor is osmotic stress, but we might in the future with this new biosensor try to prove it in different conditions, heat, pH.
Awesome. Nice. Thank you. Great. Thank you so much, Itzel.